thanks a lot for the invitation. I mean, it's really an honor and to be here. It's great. And actually, when I got an email from Tomek Włodarski, I replied that I'm not yet famous scientist, definitely. So he might have mistaken me with someone else. But then no, he was insisting that no, he meant me, and so I'm very happy to be here. The field of my research, as mentioned, is iron homeostasis, and the work that I will show you today is mostly the work from my PhD and postdoctoral time, because as a young PI, I'm getting paranoid about competition, so I decided not to show any of our ongoing work, but just some topics that we are following. But I'm very happy to discuss informally over the wine reception about what we are actually doing right now in the lab. You may wonder why we should care about iron homeostasis or about iron levels in our body. It is because iron homeostasis or iron balance is essential for health. So on one side, when you have too less iron, this will impair metabolic functions of virtually all the cells and impair growth. And in the context of the whole body, it will be hallmarked by iron-restricted erythropoiesis. So this obviously leads to anemia, and there are different types of anemias. For example, iron deficiency anemia coming from nutritional iron deficiency, which is actually a global health problem nowadays. There are also other types of anemias, for example, quite prevalent anemia in hospitalized patients, which is called anemia of chronic disease, which is basically driven by inflammation. On the other hand, when there is too much iron, it's also quite bad for us. So iron overload will lead to oxidative damage, so the ROS production, as Agnieszka was mentioning, and this will eventually lead to organ failure. This hallmarks different types of diseases again. One of them is hereditary chemochromatosis, actually very, very frequent genetic disorders, especially in our Western or Caucasian population. But there are also other types of iron overload. For example, there is too much iron accumulating in the body in a few types of hereditary anemias. One example is thalassemias. But nowadays, we also understand that iron accumulates or is misdistributed in many other types of diseases. Some of them are pretty common or frequent, like chronic liver disease, type 2 diabetes, or neurodegeneration. So actually, it's very important to understand how iron homeostasis is actually preserved in our body. And this is something that definitely you can learn, not even non-biologists definitely, how actually this is maintained in our body. We absorb iron from the diet in duodenum, and then iron is distributed throughout our body in the form bound to plasma protein transferrin. And most of iron, actually, bulk majority of iron goes to the bone marrow for the production of red blood cells. When they are getting senescent in humans after around 120 days, there are specialized cells in our body called macrophages, which are professional phagocytes, or cells that eat other cells, and they reside mostly in the spleen. And they are phagocytosing red blood cells, and they are very good in releasing iron back to the circulation. Of course, also iron goes to all the other tissues. What is very important about this kind of routes is that the excretion of iron is relatively minor, and it's definitely not regulated. So therefore, we have to really control very well both iron absorption from the diet as well as the release of iron from the stores. And this is the job of our favorite master hormone that is called hepcidin. And as hepcidin actually is like insulin for sugar. It's basically working in a very similar manner. With this exception that it's not produced in pancreas, but it's produced in the liver hepatocytes. Hepcidin is released to the bloodstream when there are conditions such as high iron levels or inflammation. And when it reaches the target cells, which are mainly cells in the guts, in intestine, and enterocytes, or these macrophages in the spleen, it actually can bind to the iron export channel, the only known iron export channel, which is called ferroportin, and mediates degradation of this this channel. So you can imagine that the iron is kind of locked inside the cells, and it's not released to the circulation that efficiently anymore. So the levels of iron in our body or in our blood drops. On the other hand, under conditions that suppress hepcidin in the liver, which are, for example, increased erythropoiesis, low iron levels, or hypoxia, there is, this suppressive function is not anymore so potent, and therefore more iron is released from the cells into the bloodstream. So you can now imagine that the diseases that I described at the beginning are actually, now we understand that they are very much related to dysregulation of hepcidin, with overshooting hepcidin responses being implicated in anemias and hepcidin deficiency being implicated in iron overload. So what is the big question in our field for many, many years right now is how actually these systemic signals, like for example iron, inflammation, erythropoiesis, are actually translated to the control of hepcidin expression, which is mostly transcriptional, molecularly. So what are really the signaling pathways, the mechanisms that translate these signals to regulate hepcidin promoter activity? And actually, when I was looking at this slide yesterday for like 20th time, I realized that actually for many years we are investigating this in many groups, and we still don't understand it. So there are still signals like iron that we know induce hepcidin, but we still don't have the complete picture how this molecularly happens. But still, this is a big interest. And when I was entering into the field some years ago, what we knew was a kind of signaling scaffold for hepcidin control, so consisting of two major pathways. One of the pathways is so-called Bmp Smart Pathway. It's triggered by BMPs. Those are small cytokines from TGF-beta superfamily. And this goes through SMART transcription factors that then sit on the hepcidin promoter, which is depicted here, mostly on these BMP responsive elements, proximal one and the distal one. And we know that this pathway involves the proteins that are normally, or not normally, that are mutated in chemochromatosis, so hemojuvelin, transferrin receptor 2, or HFE. So they act as hepcidin activators by mechanisms which, for example, for these two are still not very well understood. But we know that this pathway is important for iron responses, although we don't understand how this really happens in much detail. And we know that this pathway, the SMART factors, are somehow impaired or less phosphorylated in chemochromatosis. The second pathway is a typical inflammatory pathway, JAK-STAT pathway, triggered by some pro-inflammatory cytokines like interleukin-6, for example. And this pathway is hyperactive in this very broad group of diseases or the condition that I mentioned before, anemia of chronic disease. So coming back to our diseases, the pathways that I showed you could explain why iron is, why hepcidin is kind of too much induced, for example, in anemia of chronic disease because of the inflammation, or why it's suppressed in hereditary chemochromatosis because of the suppression of the BMP pathway. But actually, this knowledge was not telling us, for example, why hepcidin is very suppressed in thalassemias or in some other acquired diseases, for example, chronic liver disease. So we thought, okay, maybe we should learn about hepcidin regulation in more unbiased fashion. Yeah? So this was a major aim of my work. And as a starting PhD student, I started to design and optimize and establish a screening technique. I don't know if you are familiar with this, probably not, but I try to explain what's going on. So we were taking cells that are hepatic cells, HUH7 cells, and we were kind of seeding them in tens of or dozens of 384 well plates that in every well we had another siRNA. So siRNA is a small piece of RNA that via RNA interference can suppress each gene of the genome. Yeah? So we were doing this on genome-wide scale. Yeah? So in this type of assay, we were knocking down the genes in the cells, and then the cells were transfected with two plasmids. One had promoter of hepcidin, driving the luciferase, so a gene or a protein that gives a bioluminescent signal. And the second promoter that we were transfecting was just constitutive for normalization of the signal. So basically what we were learning from these measurements of luciferase activities was which of the knockdown, so which of the depletion of each gene of the genome changes the hepcidin promoter activity, either up or down. Yes? So we were trying to identify new hepcidin regulators by this way, or at least transcriptional regulators. And this genome-wide screen, so we were doing this, as I said, for all genes, 19-something hundred, 1,000. We came up with, when we applied certain thresholds for the, let's say, the hits from the screen, or when the whole library of this sRNAs was kind of plotted as a distribution of, let's say, signals. So this kind of represents suppression of the hepcidin promoter and this activation of the hepcidin promoter. We could see that many of the known hepcidin activators are actually kind of giving the phenotype as we were expecting. Oh, you cannot see actually much. One very important hepcidin suppressor that we actually identified also by some smaller screens that we did before, SMAT7, was very nicely showing a phenotype of a suppressor. They were where we would expect them to be in terms of their effects, yeah? But then when we applied certain thresholds for the kind of identifying candidates from this, or candidate genes from this screen, we found 1,600 putative activators and around 500 suppressors. And you can imagine that for a PhD project, this was a huge challenge because, well, I really I was puzzled. What should I do with this hundreds of candidate genes? So then what we actually did, this one of the approaches that also Agnieszka mentioned, was quite a bit of bioinformatics to try to look at our candidates and look for the functional enrichment, so what kind of functions of the proteins are enriched among these hits. This was one approach, and the second approach was to try to, also using some bioinformatic databases, to look at the connections, functional connections between the genes. And what I'm showing you here is a kind of merge of these two approaches with some overrepresented functional categories among our activators. So now we focus only on the putative activators, drawn as an interaction network. And what was really striking for us is that among our activators, both transcription regulators, signal transducers, and inflammatory genes were overrepresented. And what was also very interesting is that when we looked deeper into signal transducers, we saw genes that are kinases or involved in phosphoinositide signaling or in the suppression of RAS over here, and I will come back to this a little bit later, as overrepresented categories. So pointing that among these genes, something interesting could be hidden that regulates hepcidin. So taking these categories as a starting point, we decided to choose quite some genes from this network that are kind of signaling hubs, so that are important genes for certain signaling pathways to test if this pathway or this mechanism could be affecting hepcidin expression. So in this way, we chose 61 genes for like validation assays to see if they're really true, yeah? And from them, we could validate 15 genes. And then when we looked, of course, what these genes, so here are the genes, 15 genes. I will tell you in a second what they are doing or in which pathways they are involved. Some positive contours that we also put into our validation assays. And here what is shown is a knockdown efficiency, so how well the siRNA was putting this gene down at mRNA levels, and how hepcidin mRNA was reacting to this. And here what you see are kind of pooled siRNAs. So for each gene we had 4 different small RNAs to deplete them. But basically for gene to be validated, it was a little bit more complicated because for each of them we actually were using 4 separated siRNAs and a pool. And we were saying that the gene is validated only when at least 2 siRNAs were putting hepcidin down, and when this phenotype on hepcidin was correlated with the knockdown efficiency, so how well siRNA was working. So now on the table, you can see 6 of our candidates, which actually were transcription factors. And by the color code, you see how many siRNAs were doing the job, and if this effect was correlated with their potency, with the knockdown efficiency. And well, for the functions, of course, we don't go here, but they were actually quite diverse in terms of functions. Some of them involved rather in inflammation, some of them in more general responses, and some of them, like serum response factor or CREB binding protein or yin-yang 1, are typical transcription factors that are involved in assembly of bigger transcriptional complexes. They are not very specific to one pathway, but they are known to kind of interconnect with other transcription factors at many different promoters of many different genes. And the, okay. Yes? Yes. No, at this stage we were not doing Western blots. So we had too many, I mean, we were kind of working in a semi-high throughput manner still here. So we had 61 genes to validate, so we were not doing Western blots back then for them. Okay, so these are the transcription factors. And then the other 9 genes were signal transducers, again with the same color code, involved in many different processes that could be kind of summarized as inflammation, nutrient sensing, growth factor dependent signaling, morphogenesis, or even circadian rhythms. But of course you can see this is very diverse in terms of function again. So then we, I had another challenge what to do next with this data set. So we asked the question, are these candidates acting via the known pathways that regulate hepcidin, or somehow they act independently? So to tackle this question, we first took a kind of promoter approach. So you remember these 2 pathways that I showed you, the BMP and the JAK-STAT pathway, and I told you that they operate via these well-defined promoter elements, the BMP responsive elements and the STAT binding site that is very close to this BMP responsive element that is here, which is a proximal one. And what we did, we used an hepcidin promoter with mutations of this site. So this is for STAT, and this is for the both BMP responsive elements. And first I show you what the positive controls are doing in this type of assay. So when we deplete again with siRNA, hemojuvelin, that is one of the kind of upstream regulators of the BMP pathway, or STAT3, that is the transcription factor here, and we do it in the cells that are transfected with these constructs, and we measure the luciferase activity, we can see that in the knockdown of hemojuvelin, only the wild-type promoter is reacting, also the one that has the STAT mutation doesn't care, but the one that has 2 BMP responsive elements mutated is not repressed anymore, or not significantly repressed. And when we knock down STAT3, it's a bit different. The wild-type promoter is suppressed, but both the STAT mutant and the BMP mutant are not suppressed anymore significantly. So then we kind of took the same approach for all of our candidates, using the same 3 versions of the hepcidin promoter, but also 2 other promoters as specificity controls. And here what we saw is that for actually all of our candidates, apart from only 2 genes here, we could see that the wild-type promoter and the STAT mutant promoter are actually nicely suppressed when we knock them down with siRNAs. This doesn't happen for these unrelated promoters. Even we see some upregulation for some of the knockouts, but we never follow this. What was the most striking is that then we have this double mutant that doesn't have this both BMP responsive elements. Most of them are not affected. So there is a huge difference, yeah? And this led us to conclude at this stage that these two elements serve as a promoter elements that link multiple signaling events to hepcidin transcriptional control. And they are not only involved in this BMP pathway, but they are kind of conferring broader functionality of this promoter. So without these elements, the promoter doesn't actually respond to almost anything. Okay, and then another approach that we took to tackle this very similar or the same question about the involvement of the known versus completely new pathways was to look if the knockdown of these candidates that we have changes the fault induction of hepcidin in response to interleukin-6 or in response to BMP6. And when you look at two upper genes are positive controls, STAT3 and SMAT4, so the transcription factor is crucial for these two pathways. So when you see, I mean, when we knock down STAT3, the response to interleukin-6, the fault induction is much smaller. When we knock down SMAT4, we see that the response to both interleukin-6 and BMP6 is, I'm sorry for the colors, it's actually quite dark here, is also suppressed, yeah? And this is not surprising because we know that the SMAT transcription factors are kind of important for both the BMP response and the inflammatory response. So they are also kind of making the hepcidin promoter functional, let's say. And then what we noticed when we were doing the same for our candidates was that for several of these genes, we saw their contribution to interleukin-6 response. For only 3 of them, which were actually all 3 transcription factors, STAT6, CREB binding protein, or serum response factor, we saw that they mimic SMAT4, so they are important not only for the steady state expression, but also for the induction of the hepcidin in response to inflammation and BMP6. And lastly, we saw that some of our candidates are actually only important for the steady state response. So altogether, we thought, okay, we cannot really pinpoint completely our candidates to the known pathways. There must be something else there. And we want to learn more about this. So we decided to take another unbiased approach here. That was a pretty bigger one. So what we did, we used so-called reverse phase protein arrays. So those are arrays in which you can measure both the steady state levels and in some cases phosphorylation, so activation state of a panel of proteins. They were around 80, and they were proteins involved in cellular signaling, so the most common ones or the major ones that could be then grouped to different signaling pathways that are listed here. And basically what we were trying to look with this assay was what happens to this signaling network in cells, in our hepatic cells, when we knock down our candidates. And here we didn't take the transcription factors, but those guys that are acting more upstream in signaling events. And we saw that at 24 hours upon knockdown, not much was going on the signaling events in the cell. But at the 1 day later, at 48 hours, we could see that actually 7 of our candidates were provoking very robust changes in cellular signaling, in contrast to 2 other guys that were not doing much, so nothing was changing here. Similarly as for 2 our positive controls, which were hemojuvelin and BMP receptor, that again didn't have such a profound alteration of signaling events. And coming back to these guys here that are in pink, we could see that the pathways that are mostly kind of pushed out of balance upon depletion of these genes were the RAS-RAF-ERK pathway that is important for proliferation and response to growth factor, the ROS signaling, beta-catenin signaling, PI3 kinase signaling, but some other pathways were actually not really affected. So what we thought at this stage was that for these genes, a kind of sum of this alteration leads eventually to hepcidin suppression. But of course, with this type of data, we could not say which pathway is doing the job, yeah? So what really directs or what really leads to hepcidin response? So what we decided to do was to probe this signaling pathway. And this is pathways, which is depicted here in a very simple scheme, I mean, quite simple scheme. We start with an oncogenic protein that is called RAS. It's a small G protein that can signal via the PI3 kinase down to AKT kinase. And AKT kinase then has some other effectors. One of them leads to beta-catenin signaling, another to mTOR signaling. And when we go back here to RAS again, it can also signal directly via very typically pro-proliferative pathway, which is MAP-RAF, MAP kinases pathway. And this goes eventually to many other effectors, but mTOR is one of them as well. So with this kind of scheme that basically was represented in our arrays, in this reverse protein arrays that we did before, we decided to probe this, this network either by small molecules, so by inhibitors of certain hubs of this network, or by overexpression of the crucial components of this network, mostly the oncogenic or the very active versions of these proteins. And all this, of course, in the same hepatic cells that we all work all the time with. And of course, the data from this type of testing was pretty extensive. And here I would like to just pinpoint to our most important observation. So our most important observation goes here in the RAS-RAF. So what we saw is that when we used sorafenib, that is a RAF kinase inhibitor, we could see very nice induction of hepcidin mRNA. And when we used the hepcidin promoter as a readout, or its activity as a readout, and we overexpress in our cells the oncogenic version of RAS, or the BRAF kinase, wild type, or the BRAF kinase, especially the very active variant of this that is very oncogenic, we could see that hepcidin promoter activity is very much suppressed. And we didn't see this for another more downstream MAP kinase, which is MEK1, showing that this is somehow specific to this most upstream events in the MAP kinases pathway that are suppressive to hepcidin. And then to kind of give some physiological importance to this data, we established collaboration with a group that had conditional knockout mice for one of our favorite candidates from the screen that we validated and that I was showing on previous slides, that is called RASA1. RASA1 is a RAS suppressor, so GTPase of RAS that inactivates the RAS. And these mice were done in this way that they were inducible. So the mice have to be injected with tamoxifen to delete the RASA. Yeah? So we used these mice and we injected tamoxifen for 6 weeks, and we saw that in the liver, already after this relatively short time, hepcidin expression is suppressed, which led to some alteration of systemic iron levels. They are not significant, but still the reviewers were very convinced about this, because these both are indicative of this suppression. So we have a trend towards higher plasma iron levels and somehow lower iron content in the spleen. So this suggested that components of the RAS signaling could be candidates for modifiers of systemic iron levels in vivo. So to conclude this part, I would like to tell you that our screen provided the first list of potential hepcidin regulators. Of course, it's not a kind of final data, but it could be used as a good resource with combination with other bigger data sets to identify genes involved in iron homeostasis. or modifying iron homeostasis. And from more mechanistical point of view, we found a few very interesting transcription factors that some of them are also very important for hepcidin response to BMPs like STAT6 or CREB binding protein or SRF that we speculate, and this is one of the things that we follow in the lab, could kind of help to build a bigger transcriptional complex at the hepcidin promoter, maybe involving a kind of looping between the two BMP responsive elements. It was never actually proven, and this is something that I would say it's very worth to work on. And from, let's say, more upstream perspective, we added one more pathway that is suppressive to hepcidin, which is pathway initiated by RAS and RAF MAP kinase, which is important for proliferation and response to growth factors. And we know that this pathway is activated in different liver stress conditions, such as alcohol liver injury, hepatitis, or liver cancer. And we knew from clinical data that all these conditions are associated with hepcidin deficiency, but only, I mean, not only, but our observations from our cell-based work and mouse models showed that this could be one of the pathways responsible for this observation. Okay, and now I would like to switch for the last few slides towards a newer project that was also initiated by the screening data that was a search for hepcidin modulating therapeutics. So this project was initiated by some interesting observations or interesting facts. Is that first of all, there are already a lot of proteins, a lot of molecules that are approved by FDA for therapy, and they are now around 1900. And at the same time, how hepcidin is regulated is getting more and more complicated. I mean, there are several signals, apart from those that we knew for years, that affect hepcidin. So, and for some of them, we still don't know really the exact mechanism. So this altogether could suggest that there are probably several approved therapeutics that may affect hepcidin. And this could have two consequences. First, that some of these drugs could be maybe used for drug repositioning. So the approach to use the known drug for new indication, for new type of disease, which in this case would be iron-related disease. But second implication that I personally find even more important is that there could be iron-related side effects of actually quite some approved drugs that we have in the clinics now. So to tackle these questions or to tackle this issue, we decided to use our screening data to look for genes first that would be drug targets. Yeah? So our starting point was again our screening data, and then we undertook this very kind of systematic approach in this project. So we started with our hundreds of hits, and we applied bioinformatics again to now look for drug targets. And for this we used some databases of drug targets, and from this we could nail it down to more than 100 screening hits, potentially sensitive to therapeutics. From this, then we were doing a lot of manual curation to find those that make some sense, that they seem to be at least to some extent specific, not, for example, metabolizing a drug only. And from this 56, we then tested which of them are actually expressing our cells, because this is important. And we went down to 37 putative potential drug targets that could be hepcidin regulators. Yeah? So then we took first approach to validate this gene with this pooled siRNA, so the pool of 4, to see on hepcidin mRNA level if we could anyhow recapitulate the screening phenotype. By this, we could nail it down to 13 genes, and then we decided to split. So we were knocking them down with then single siRNAs, like in this similar approach that I showed you some slides ago. But at the same time, we used the database knowledge to then choose the drugs that correspond to these genes and to use them in cell-based assays first in our cell line. And actually, here are the genes the 13 genes that we tackled with assays and the corresponding drugs. I mean, you don't see much again. We were basically testing first in HUH7 cells if these drugs are affecting hepcidin. We basically then found the drug approach much more informative or much more clinically important, so we kind of focus much more on here, or at least I focus on this for here for the talk. And from this we found five drugs that were spironolactone, diclofenac, imatinib, SAHA, and amiloride that were affecting hepcidin HUH7 cells, and then we were basically testing these drugs further in primary cells, in primary murine hepatocytes and human hepatocytes, and then we kind of could confirm 4 of them. The 4 that are here: spironolactone, diclofenac, imatinib, and SAHA, which is the HTAC inhibitor. And then basically what we thought of doing, so okay, we had 4 drugs. I don't want to tell you now what they are doing, because we decided to take one more approach to see if they are really doing something to hepcidin, was to just take mice and treat them with these drugs in drinking water. And we chose the doses of these drugs that were corresponding to those found in patients and were not toxic according to FDA documentation. And we treated the mice for 2 weeks with drinking water. And what we could see is that only 2 of them, the imatinib and spironolactone, were suppressing indeed hepcidin expression in the liver. And this could not be linked to any of the liver toxicity, which normally we know can suppress hepcidin, so the drugs were not toxic to the liver. And for both treatments, we could see significant, although not very strong, induction or rise in transferrin saturation, suggesting that the iron status of the mice is somehow elevated. And now I tell you what are the drugs, because I didn't want to give you too much of the information before for those that we could not finally validate in vivo. So imatinib, its another name is Glivec. It's a pan-tyrosine kinase inhibitor that is used in certain cancer types, for example, chronic leukemias. And this is a first-line and lifelong therapeutic, so it's used for a really prolonged time. Here we kind of we proposed in the paper that the excessive iron accumulation that could happen upon long-term imatinib treatment may be actually deleterious for cancer progression, because of course data are a little bit mixed, but still most of the studies show that higher iron status is actually kind of increasing the risk of carcinogenesis or worsening the prognosis of cancer. So too much iron is not good for cancer patient, definitely. And the second drug is spironolactone. Is mechanistically, it's a blocker of aldosterone receptor. And what is important, it's used for now, let's say 50 years, I think, as an antihypertensive drug. That is also prescribed for edema, for a condition where we accumulate too much fluid in abdominal cavity. And this is very often found in patients that suffer from the heart failure, or also who suffer from liver cirrhosis. And we know that at least in liver cirrhosis, there is already quite, I mean, it's a condition of iron accumulation, and that is also associated with quite low hepcidin. And we speculate that in this condition, when we put this drug that also puts hepcidin down by itself, if it happens in patient, then iron accumulation, further iron accumulation may really worsen the disease. So altogether, our study showed that, or really provoked a need for evaluation of iron-related side effects for these 2 drugs in patients who take them for a really long time in both cases. Okay, so now I would like to summarize and tell you that in my previous work I was using both unbiased and some biased approaches to tackle hepcidin regulation. And now in my new lab, we kind of follow this, this interest, but in a broader sense, and we look in iron sensing in general. And I'm very happy to discuss which genes and which mechanism we are following here. When iron sensing is kind of failing or it's overridden, then too much iron may be accumulated in our body. And in another project, we are actually trying to look for mechanisms that are important for iron uptake, especially of this excessive iron, and especially in the liver. And in our third area of interest, we are looking at iron homeostasis in macrophages. So, and I'm happy to discuss this further. One of the things that I want to point out is you saw more or less what type of methodologies I was using before, especially for the genetic screen. I would like to tell you that in Poland, I'm trying to do now things even more advanced and more modern. So one of the things that we are now establishing in our lab and working on is a kind of new approach to dissect regulatory networks of your gene of interest. So in this, in our at least one or two of our projects, when we are interested in regulation of XYZ gene, what we successfully did already was to tag this gene in a, let's say, in the genome, so endogenously, with a fluorescent protein in a way that the endogenous promoter or endogenous regulation of this gene could be then read with the fluorescence signal, for example, in flow cytometry. And we use CRISPR-Cas9 system to do this type of knock-in. Yeah? And then what we will do, we haven't done it yet, so we want to use an approach of so-called pooled genetic screen with, again, the CRISPR, so with sgRNAs. Those are small RNAs that you may have heard about CRISPRs, yeah? So which are kind of mediating the cleavage of genes in DNA, leading to their knockout. I don't want to go into any details, but basically it's a technique to also deplete or knock out genes of interest. And we again would like to do it in a genome-wide scale, but in a completely different approach than I was showing you before. Instead of using dozens of plates, we want to have some of the big dishes with cells that we deliver these sgRNAs with viral vectors in a way that one cell gets one virus that carries only one SG. So one SG means one gene was knocked out, was depleted from this cell, yeah? And then after selecting cells that are in fact infected, we would apply FACS sorting, so sorting of cells according to their fluorescence signal. So then we would sort two populations that have very low fluorescence or very high fluorescence, and we would compare them to cells that are not sorted at all, using the next generation sequencing of these sgRNAs that got incorporated in each of the cells that we sorted. And in this way, by applying some computing, we would look for enriched sgRNA, so enriched knockouts in these extreme populations of cells, comparing to unsorted cells that in principle should represent the both activators, putative, or suppressors of any of the genes of interest that you would like to learn about the regulation of this gene in an unbiased fashion. So I'm very happy to explain a bit more this approach, which now seems to be really a new direction in functional genomics, and one of the things that we really try to do. So with this, I would like to thank my lab in Heidelberg, especially my bosses, Martina and Matthias, and my colleagues. I would like to acknowledge my growing small group in Warsaw, in the institute that Agnieszka was also working at for some years, which is also very nice, and we are also looking for students, postdocs, and PIs, so please don't forget about us as well. I would like to thank the collaborators, the funding in Germany, but especially also I would like to acknowledge the very nice grant that I got from National Science Center that is called Polonium, which facilitated my move back to Poland, and which is also a very nice program for different types of training, and we are really taken taken care of very well by this fellowship. So thank you for your attention. I'm happy to take questions. Actually, I wanted to answer the question about the Western blot. For hepcidin, there is no Western for hepcidin. If you were wondering about hepcidin, it's I mean, there is ELISA that is very expensive, and I'm actually pretty sure that it works mostly for mouse hepcidin, and we mostly use it for detecting hepcidin in mouse serum when we expect that it might be affected. But definitely there is no Western blot assay for hepcidin, which is actually quite small peptide. Hepcidin is actually regulated pretty only transcriptionally. So the levels of the peptide in the blood pretty well follows the mRNA that we find in hepatocytes. So it's very tight connection that is actually quite striking for a gene of such an important function, that it's not regulated post-transcriptionally or in other way. So it's very, and the promoter has all the answers.
So thank you for the very interesting talk. And you mentioned that hepcidin is secreted in low iron levels, but also inflammation. So I was wondering, what is the role of this hormone in inflammation?
So hepcidin is released in high iron levels. In low iron levels, it will be suppressed. And inflammation, yes, indeed, it's released. So it's it's activated transcriptionally, and then more of it's released. And and the reason for this is actually that when hepcidin is high in the bloodstream, it kind of mediates iron restriction, mostly in the splenic macrophages. So there is so iron is kind of stopped in the spleen, and the hypotheremia, so the lowering of iron levels in our serum progresses. And this has one consequence, that the microbes have less iron to survive. And the other thing is that, and this is quite intuitive, but the other related mechanism is that when we lower the transferrin saturation, because that's what happens, the transferrin that is in our blood has less and less iron, it's more free to bind iron that may appear in our body here and there because of tissue injury, because of the invasion of the microbes. So when there is some tissue injury, there might be some free iron, so not bound to transferrin, released. And this free iron is very dangerous. So the lowering of transferrin saturation also kind of released the transferrin that could then catch all the excessive iron that may appear at the site of inflammation. So two mechanisms, but altogether it's to deprive pathogens of iron.
Just a short technical question. In one of your last slides, you showed that you had 37 targets that you wanted to, from the siRNA screen that you wanted to verify, of which you only 13 proved to be really...
So from 37, we went down to 13 because the original screen was based on luciferase assay, so we were measuring hepcidin promoter activity by, in response to knockdown with these 4 mixed siRNAs in the library. And then in our first round of validation assay, we were taking the same 4 siRNAs and measuring hepcidin mRNA level. And then when we could not confirm, even with this, the phenotype from the screen, we were kind of saying, okay, this gene is definitely not validated. Mm-hmm. And then for this gene that were validated by this first type of assay, we were then deconvoluting them in these 4 siRNAs to see how each of them is behaving. Mm-hmm.
So, because I was just curious if this is typical from screens that like 1/3 maybe of targets would be possible to verify, or is it just
so
because
validated
you had
and
the different...
very first level, yeah? So
between the 37 and the 13, yes,
but
this
from the 13 we went then to
of course you
5,
went down, but
yeah, that we believed much more, because then at least 2 siRNAs. So in the siRNA field that is fortunately passing kind of over, and it's getting less and less used in screens. The golden standard was that if 2 siRNAs that are, of course, not overlapping, give you the same phenotype, it's okay. Yeah, you can see, okay, I have my phenotype, it's specific. Of course, in my data, I very often saw that, okay, 2 are giving me the phenotype, 2 other are giving exactly the same phenotype, They are kind of exactly in the same way efficient, but they are not giving me the phenotype. Yeah, so then I was rather saying that this gene is not validated.
So if I rephrase it otherwise, if we see some screening results, you know, some big siRNA screening results, should we kind of expect that maybe, you know, just this very small proportion is going to be true?
I mean, screen is always a screen, so definitely the validations that I was showing in some of my slides is just a first first phase of the project. Yeah, it doesn't mean that if you then say, okay, my gene is validated, then it's the end of the story and it's kind of functional for all the models that you might be using and so on. So then I think then the really biology starts. Then you take this gene and you try to tackle them from different angles. Then you use so I mean, what I think right now when I got older and more experience, I think that very important thing would be to validate your genes in primary cells and in mice. So basically to take it further, yeah, not to stop at the level of cells and so on. What we did for the story that I showed as the first one was rather to go then via this reverse protein arrays towards other signaling pathways. So we were not focusing that much on candidates anymore. We made come back to them and then use some additional work to really pinpoint their function. But what we then moved is rather to let's say, more broader signaling events that may affect hepcidin. So basically, it's important to take it somewhere farther. Yeah, I mean, the screen by itself is not a story. Definitely, like I heard from structural people that finding a structure of a protein is not yet the paper as well. So you have to find then a context, yeah, and you have to make a story out of it.